title. Now I want to show you quickly about this tool that is available, and you can use it for performing network analysis. Right, so let me show you. So this tool called Cytoscape, it is very popular, and it has been very widely used in many research papers that have been published, and it is very you can say regularly updated, and a lot of people or whole consortium from many institutes, and they are working on it, and it is open source, and also you can work as a developer, so you can submit your own code and packages, apps nowadays, not packages, apps can be submitted that can be installed, or they can be used in this tool to solve any biological question. So this is mainly developed for biological purpose, but now other people from other fields are also using it. And of course there are other tools like Jeffy, but that is not meant mainly for biological networks, but Cytoscape is more suitable when you are doing this network analysis in biological networks. So these are just one example biological networks. You can see these are different genes in the node, and then you can see there are different connected components, not components but subgraphs that exist in this network. And this is just an example, so you can also think about it in terms of other social networks. So what is Cytoscape? So it is an open source software platform, and it has many applications ranging from data analysis, visualization of molecular interactions and biological pathways, and also it can be used for integration of networks by including with their annotations, and you can also perform gene expression profile integration. So it is very useful tool when it comes to solving biological problems, and various applications or in form of apps are available to solve individual problems as well. Like for example, integrating the gene expression profiles, there is one app. Then studying the disease networks, there is one app. And then for studying protein-protein interaction, there is a separate app available. So depending upon your requirement, you can install the particular or corresponding app, and then you can use that app for doing the analysis. So as I was saying that this is primarily designed for biological research, but now people from other fields are also exploring this. So the core distribution of this Cytoscape, it comes with a basic set of features, and different additional features are also available in the form of apps, and formerly known as plugins. And now, as I was saying, that apps are available for network and molecular profiling analysis, or new layouts there. And also additional file formats can be supported with the help of these apps. Then scripting and connections can also be built with the databases, can also be established with the help of these particular apps. Now just a brief version history of this tool. So it was initially released in 2002, almost 2 decades ago, and it has undergone numerous development sprints. And it started with the version 2.6. It's a legacy version now and this version is no longer existing. And as of May 2016, there has been transition from this 2.x series to 3.x series, and 2x is no longer supported. And now latest version of this Cytoscape is 3.x series, which is I think the latest version now also is 3.8.2. It is written in Java programming and also required the Java runtime environment to be installed. And it has new features, including it has new interface now, it has advanced visualization functions, and it is one can also use command line distribution. And there are also many apps like RESTful APIs and multiple rendering engine support. These are all features of this new 3.8.2 version. So how do you get it? So simply you can go to your browser and you can type cytoscape.org, and you can quickly you will fall on the front page, and where you can just simply see that the first option is to download this Cytoscape. Should I probably take you through quickly? All right, just need to change the screen. Excuse me for that. Okay, so I hope you are able to see it. You can go to this URI, that is cytoscape.org, and then you will come to this web page, right? So from here you can just download it, and yeah, so latest version is 3.8.2, and once you have installed it, then you are ready to go to use it. One issue might be that you probably have to either update your Java runtime environment. I think it is the requirement for this tool is I think 11.0 onwards. If you have not installed it, then you can just then it will be by default installed by this Cytoscape tool. So those who are interested, please install it. I can wait for 4 or 5 minutes, so that you can also try to have some hands on it, very quick hands on. So after installation, you will when you open your Cytoscape tool, you will see this kind of interface. And let me just take you through this interface quickly. So it has this bar called menus, where you will find all the options for menu, and like file, how you can read the file, import the file, sorry, or how you can generate the new networks, and so on. So, and also you can, if you want to install apps, you can click on this, and then you will get to the new panel where you can, and I will show you also in the upcoming slide how you can do it. And then you have a list of tools that are available, like one of them is most important one is network analyzer, where you can calculate all those network features such as betweenness centrality, or closeness centrality, and degree distribution, degrees or not degree distribution also, yes, and so on. And then you have this toolbar where you can have quick functions like import from table, import from SIF file, import networks. You can save it or you can open folder and so on, zoom in, zoom out, and so on, right? So there are many functionalities that are quickly accessible. I think the most important thing in this panel is this network layout panel, sorry, this network panel, where you can look out for networks that are biological networks that are deposited into different between different deposited in different databases, data repositories. And a few examples are this string database, where you can find this protein interaction networks, and then there is also this NDE, where you can find many different kinds of gene regulatory networks. You can find metabolic networks and also other kind of gene protein interaction networks or RNA protein interaction networks. So these are all different kinds of network that you can explore. So you just type in over here. So I will also quickly show you how you can do it. And the next thing is this a network view panel, where you will so this is by default. When you come to the starter panel, you will see these example sessions that are provided. So even you can just for exploration, you can also try to just click in on one of these sessions, and this will give you some demo or you can say dummy or already default networks that comes with this tool. And this is also called this this sample or network view panel, where you, if you click on one of these, you will see the network. So there are many examples that have been already provided as examples to this tool. So for example, this affinity purification network is provided, then yeast perturbation network is provided, and then there are different layouts to compare how different layouts can be used. So there is in the menus, you can see there are layout menu, where you can change the layout of your network. There are so many interesting layouts, visualization layouts that are possible. So you can just pick which one you find suitable or interesting for your purpose, because sometimes you might have to let's say if you are looking for degree distribution, probably you will prefer to have your network organized by some sort of let's say degree distribution, right? So that is possible. And you can also have circular networks, so you can have hierarchical networks, or you can have this tree-like structure for the networks. So these are all different layouts that are possible. And the next important thing is this table panel. So corresponding to each network, you will find a corresponding node table, ads table, and the network table. So you will find the details of all the nodes, like the names, labels, and things like that. And if you click on the ads table, you will find the details of edges. So what is the interaction strength, what is the direction, and things like that. And network table also some information about the net. And you can also, I think, create new tables. That is also supported in the new version, I think. And then you can have this style tab. So below the network, you can also go to style, and you can change the labels, color of the nodes. You can change the shapes of node. You can change the color of the edges and things like that. And another important one is filtering. So suppose if you are interested in let's say looking at only those nodes with the particular degree, right? So you can filter and you can put a criteria, and you can filter all those nodes which have degree, let's say greater than some threshold, or greater than or less than some threshold. So these are all possibilities. You can filter the nodes or edges, right? So these are most of the time very useful requirements. Then you can also, I think there are apps now for annotation, so you can also use that. And there are also layout tools. And then there can be many other, depending on what apps you have installed. And yeah, so as I was talking about how you can install the apps, so you can follow this URI apps.cytoscape.org. So this, you will come to this web page where you will see that there are many useful categories. So for example, you can have apps that are suitable for data visualization, or you may have apps for data network generation, right? So you can have network analysis apps, you can have online data import apps, you can have systems biology related apps, you can have apps that perform clustering, that perform enrichment analysis, that perform integration, that perform annotation, that those perform an ontology analysis, and so on, pathway databases. They are broader categories of these apps. And so there are two possibilities: either you just download it from here and then you just install it, or other and also like these are rated, so how many users have used it, and the users can even rate about the quality of the apps. So these are very interesting features that are provided. And also if there are any apps that are more frequently downloaded, so these are all highlighted here in the this page. And what I was saying is how to install. So I think the easiest way is that you follow up this, you just click on this apps in the menu tab on the menus, and then you will see that there is a new small panel that's popping up, that is drop down menu is app manager. You click on this and then a new window will appear, and then in this you can just type in the app name. So these are all in there. You will see all listed apps are available, and just type in the name, and then you can then select from here, and then you can just install it by clicking this button. So it is fairly very simple and easy to use tool. So you can also have a look on which apps are installed in your on your tool. As particularly this tool is very useful those who do not have any programming language or who comes from core biology background. So this tool can be used to do or to perform this network analysis. Very useful tool. And to show you one example, so I was talking about this string database where you can find this information on protein interactions. So you can install this string app. So and this will allow you to do many things. First thing is that you can directly import the networks from this string database into Cytoscape. Another thing is that you can also search by different criteria, different queries. For example, you can search by disease, you can search by organism name. So let's say if I search for this ovarian, or let's say breast cancer, so just in the search box you have to type in breast cancer, and it will fetch the corresponding network that for this particular disease. So I think I can show you a small demo of this. Let me show if there is anything interesting left in this context. Yeah. Okay, so yeah, so I think I can show you this quickly. Cytoscape, okay. Okay, I hope you are able to see this Cytoscape.
Yes, sir, visible.
Okay, so thanks. So I have already installed this app, sorry, this tool. So I also have already installed this string database, string app, sorry. So as you can see that this is already installed, but you can also install it by just typing in over here string. So you will see this app since it is already installed, so I don't need to install it. So you can also install it, and then yeah, so here for example, so you can select different query ways to query. So either you can select, if you know the protein name, so you can select this, and in the search box then you have to type in the name of the protein, or you can protein name or list of protein, right? So these are all, and then you just control enter to search, right? Or another way is you can search by the name, disease, sorry, disease name. So for example, I have already installed it for ovarian cancer. So let me see if there is any network that available for breast cancer. So you just hit it, and you see that something is going on. So this is fetching the network from this string database. Okay, so it says that it has identified 100 nodes or 100 proteins, you can say, because this is a protein interaction network. So nodes here are proteins, right? So how many proteins are there? There are 100 proteins that have been found to be kind of, you can say, associated or have been found this the involvement to this breast cancer disease. And you can see the number of edges here. So there are around 2000 edges that exist in this network. So these are possible connections between that connections have been, or interactions have been observed in these 100 proteins. So how do you now calculate these properties? So what you can do is, first thing is that you can even look at this by zooming. So control plus, you can just zoom in this network, right? So very you can say pleasant way of storing or representing the information, visualizing the information. So each node, remember, is a protein. And interestingly, if you look, so what you see inside a node, so these are the protein structures. So amazing, right? So if there is protein structure information is available, so this information you can also get. So for example, for this protein map 3K1, so there is protein structure is also available. So you can quickly also know this information, right? So for if there is no structure, this will be blank. You can see. So this is how this protein interaction network looks like, and you see this is very complex network, right? So and there are now, so there are different ways you can change the layouts. For example, as I was saying, you can change the layout by, let's say, by degree. Yeah, sorry for interruption. So yeah, so I was saying is you can change the layouts. So you can, for example, have a degree sorted circular layout. So now this network is sorted by degrees. So at the bottom you will find all those nodes with the node with the minimum degree. I think you can also see it somewhere in the table. No, we have to calculate, I think. So far we don't have this information. Yes, so for this we don't have. Okay, so how do we calculate it? So this is now already sorted. So you can calculate the degrees and other network properties with the help of this tool, and then analyze network. And it also asks whether you want to treat it as directed graph or undirected graph. So let's treat it as undirected, because protein-protein interactions are undirected graphs, in fact. So you just click on this, and now I think you will see some these properties are calculated. So you will see that summary statistics is provided. How many, so what is the network diameter, what is the characteristic path length, clustering coefficient, and how many connected components it has, and things like that. So these are all summary statistics, and also you can find these other properties like node degree distribution, betweenness, centrality distribution, and things like that. You can click on this, and I think you will then you will get it. A new tab will open, which I hope you are able to see. I'm not sure. So if you click on this, a new table will open, which can give you the distribution of this degree and all other parameters. All right, so I hope you will be able to explore this by yourself, because the time is limited for this. And so yeah, if you have any queries regarding this, you can always contact me. And I think with this, I would like to, I think, stop here. Just give me 1-2 minutes more. Okay. All right, so this was about the string app, and then there are different ways you can load the network data, and I'm going to skip this. There are also ways to perform this sub-network analysis and things like that. So so I think I can stop here. So please let me know if there are any questions.